Welcome to Careers in Discovery, your window into the world of biotech leaders. On each episode, we interview the industry's leading executives to understand how they built their careers, what they've learned along the way, and the advice they'd give to their younger selves. This week on Careers in Discovery, Emma's joined by Jonas Dyhrfjeld-Johnsen, Chief Development Officer and Managing Director at Acousia Therapeutics, who are developing innovative therapies to preserve and restore hearing. Originally a biophysicist, Jonas shares his journey through the biotech sector, emphasizing the importance of adaptability and continuous learning. Stay tuned for an insightful conversation about a major global health issue and a fascinating career.
So welcome back to Careers in Discovery. This week I'm joined by Jonas Dyhrfjeld-Johnsen, CDO and Managing Director at Acousia Therapeutics based in Germany. Welcome, Jonas.
Thank you very much, Emma. Thank you for the opportunity to join you today.
It's a pleasure to have you. I suppose a great place to start is to talk a little bit about the work that Acousia Therapeutics are doing to combat hearing loss.
Yeah, so we are fully focused on only treating hearing loss, which, at least in our experience, is somewhat of an underestimated problem by most people, both in terms of prevalence and the impact on patients and also the need for solutions. Worldwide, there's about 1.5 billion people suffering hearing loss these days, and the WHO a couple of years ago predicted that was going to rise to 2.5 billion over the next about 20 years. So it's a massive health problem in that respect already, and it does impact a lot of things in people's life when they suffer hearing loss. It's not as simple as not being able to detect a certain level of sound. It's really an impact on ability to communicate. That's what people are complaining the most about as in their daily life, the ability to understand other people, not to detect sounds, which in turn can impact mental health. It leads to isolation, leads to some degree of self-isolation when people feel that certain environments are too challenging, be that work-wise or socially. And it's even been linked now to a certain predisposition towards early onset dementia or Alzheimer's disease. So it's an in the moment problem, but it's also a long term risk factor for down the road as you grow older.
Yeah, well, that's interesting to hear that it's the rates of hearing loss are increasing so much. What sort of evidence or what understanding is there as to why that's the case?
But there's a lot of discussion around the impact of an increasingly noisy environment. We have all of us a lot of the time earbuds in our ears, increased sound exposure, increased noise exposure in out and about in the world. Probably a lot of people will notice that these days when you go to a restaurant, even there's a lot of background noise as well. So on one hand, this can contribute to making things worse. It can also contribute to emphasizing the problem once you have lost some hearing.
Yeah.
And interestingly, in terms of the solutions, most people are well aware of the possibility to get hearing aids, or even in very severe types of hearing loss to get a cochlear implant.
Yeah.
But patients actually do not feel that they benefit as much as they need from a hearing aid, and that has to do with the mechanisms of hearing that. In the vast majority of cases, in terms of acquired hearing loss, so not congenital hearing loss, but hearing loss that you suffer during your lifetime, it's the sensory hair cells, the outer sensory hair cells in the ear that initially encode sound that are affected. And the most vulnerable types of these hair cells are the ones that encode the highest frequencies that we're able to hear. And hearing aids have a limitation. They can, even the most advanced hearing aids only go up to about 10 kilohertz. And for reference, human hearing goes all the way up to 20 kilohertz. So this sort of technical limitation of the hearing aids means that first of all, they amplify sound, so they also amplify background noise, but they only amplify the part of the sound spectrum that people lose access to the last. Whereas in the beginning, they're missing the high frequencies or the very high frequencies that play a large role in speech recognition. They help you distinguish between consonant sounds like D and T and G, and that is very difficult
really
in a noisy environment. And they also help you sort of with appreciating music, because these high frequencies are also where the harmonics are in music, and that's what patients report that they don't feel is being addressed. So we're trying to preserve these cells. We're trying to boost the function of those you still have left, and we're doing that both for acute types of hearing loss with a program that's currently in phase 2, and for chronic progressive types with a late-stage preclinical program.
An amazing journey. So talk to me a little bit. You're wearing 2 hats, Managing Director and Chief Development Officer. What does that entail?
Well, Acousia is constructed in a very particular way. So we're essentially a 2-3 person outfit. We have 3 people on payroll, but 1 person is a scientist that's seconded to the local academic group that we collaborate with. A way of sort of boosting that collaboration. So on a day-to-day basis, it's really a virtual biotech. Where we split responsibilities between 2 managing directors. My colleague wears a double hat of CEO and CMO, Chief Medical Officer, and I manage the development aspects, which in our situation covers anything preclinical, translational, as well as contributing to the clinical development as well as needed. But obviously, with that small of a company, then that also means that we are both heavily involved in business development and fundraising, in anything external facing, anything administrative in-house is divided up depending on responsibilities between the 2 of us as well. So it's really a do-all situation.
Very broad remit.
And with a very lean in-house setup, which allows us to invest the vast majority of the raised money into high-quality service providers, high-quality consultants to supplement our knowledge and skill set. So small back office and put all the money into R&D.
That's fantastic, and quite quickly, as you said, you've been able to get to phase 2. So it's definitely a model that works.
Yeah, I mean, it's the target identification, and we work on what's called the Kv7.4 potassium channel that's highly expressed in the inner ear, a biologically validated target that's known to be implicated in various types of hearing loss. And that was selected late 2017. So really, in a very short period of time, the company has managed to take that all the way from early preclinical validation up through the development phases and through a successful phase 1 and now running a phase 2.
It's fantastic. And I think that leads on to a good point of your earlier career journey. How did you develop the skills? What particular fundamental learnings have you had to bring you to where you are today?
Well, I started out in basic research, and originally came going far back from physics and biophysics. Always was interested in working in the biological systems, but liked sort of the rigor and the ability to distill principles and models from physics. So the biophysics was kind of an interesting angle to get into that as well. But I must say that originally I was definitely not considering a drug development career and not even an industry career back in the day. I wanted to explore, I wanted to learn, I wanted to discover and understand. I've always felt very closely aligned with Richard Feynman's book, The Pleasure of Figuring Things Out. That's sort of just a great motivation to do more and push forward in general. So that's what put me on the path of science to begin with. But then I started out with more computer-based, biologically realistic modeling to understand single cells, to understand brain networks. That was part of my PhD work with Dr. Rolf Köser in Düsseldorf, and then from there on learned that there wasn't always the data that I needed to build these models. I was not an experimentalist at all. But got an opportunity for my first postdoc to go to the University of California, Irvine, in the lab of Ivan Soltes, who was there at the time to learn to do patch clamp recordings, to have the ability to generate myself the data that I was missing
in
the computational modeling, or not necessarily have to wait for it or convince somebody else of the importance of the model I wanted to build, but also continue doing modeling there. But that brought me sort of a step closer to medical research, to applied research, because that lab was working on epilepsy research, some aspects of basic structure function in the hippocampus as well, but really mostly driven by themes such as pediatric hypothermic seizures, so febrile seizures, that can be a problem in neonates in particular. And then also post-traumatic epilepsy, so how things changed on the cellular level and on the network level, and how we could better understand these mechanisms. So that sort of drew me a step closer to applied research and to the medical field, and I continued with that in the second postdoc in also an epilepsy lab at Harvard Medical School. And there, even as it often is, by somewhat of a coincidence of discovery, ended up in a project that developed a in vitro screening model for drug development. We were able to demonstrate that brain slices, or slices of the hippocampus in particular, actually undergo this very similar progression of generating electric seizures in vitro as you could see in vivo, but in vitro on a much shorter time scale, and obviously both from ethics perspectives, from a yield perspective, you could test a lot more compounds if you can slice the hippocampus into a large number of slices and test on all of them. Right. So that sort of reinforced the idea that it would be interesting to do something that at least maybe, I mean, sometimes in basic research, you feel that there's a very long time scale until you see a direct impact of what you're doing. And that's not to say that it isn't important, because all of those of us who end up in applied research are standing on the shoulders of those who have done the basic research to begin with. But I became interested in the opportunity to do something more immediate in that respect, and then through personal connections got the chance to join a startup, Sensorion in France, and get into the industry side of things, but at a stage where the company was very young and still had to be built.
Okay, that's quite an experience, fresh out of postdoc.
Yeah.
What qualities do you think are important when you're coming from the academic environment and moving straight into startup biotech mode?
Well, I think I don't know if it's necessarily just for making that switch or in general, but I think you need to be willing to combine approaches, both what you already know, take on the challenge of learning new things. Something that I think I already did in the academic environment by combining biology and physics, by combining modeling and electrophysiology and so on. So maybe I had some sort of predisposition for making those sort of combinations already. But you sort of have to come with a certain skill set, but you have to be also willing to change the way you do things. There's a certain level of emphasis on other things. You can't expect just to keep burrowing in depth in one direction because you keep finding relevant new information. There's other milestones to be achieved. There's other priorities in terms of sometimes needing to accept the fact that reproducibility and running the same or similar model or experiment multiple times in a row has a higher priority than chasing that one interesting data point that might take you further down the rabbit hole, right? So I think that's something that I learned across the years, that some people don't appreciate so much. On the other hand, other people appreciate the challenge of refining your models to make them reproducible, to be able to generate that robust and reliable data that enables or gives the confidence that you can take a program further, that you're not sort of, as we discussed earlier, needing to pivot because you can't reproduce your data or it doesn't work. Well, a lot of the time when things don't seem to work, it's because the model isn't stable or the model isn't robust, right? So different priorities, also a lot of different areas that you don't necessarily learn in academia, pharmacokinetics, toxicology, drug manufacturing, quality aspects of chemical manufacturing and controls. Not that it's my specialty, but it's something that I'm responsible for and oversee with the help of expert consultants. But there are things that you're not necessarily aware of when you come from an environment where research is the focus, because these things become important later down the road in development.
Yeah.
So keep the strong.
So that's curiosity almost, isn't it? Not just curiosity in the science, but the curiosity in the unknown and the what's next.
Curiosity and willingness to accept that in the beginning you're not going to be good at it, right? I mean, you come out with a PhD, you have a title to your name, you have some few years maybe of experience behind that. And all of a sudden you plant yourself in a field where the priorities are different. So it doesn't mean that your skills are not relevant anymore. Part of being in science and studying in the direction to become a researcher is learning the ability to learning the ability to change, adapting to even in research, adapting to unexpected results. But here it can sometimes be your whole skill set that needs to be adapted if you want to continue moving with the company, because the company is going to change over time, right? You won't be in early discovery, at least in a small company. Hopefully for that long.
Not. Yeah.
I mean, if you want to be specialized in that direction, but be in industry, you probably need to look at a larger organization that has a team or department dedicated to that. But if you're in small startups, the company's going to change and grow. The company structure and organization might change. So, but at least the focus and the priority will definitely change from early stage target discovery or target validation to all the other aspects in order to take a drug candidate to clinic. And if you want to be part of that, you have to keep moving. You have to keep learning.
Yeah, absolutely. And you touched on elements of your life there. You studied in Denmark and Germany. You've worked in France, Belgium. You did your postdocs in the US, and now you're in Germany. That's quite some experience.
Yeah, I mean, I think I've always tended to see it as a great opportunity in science that you have the chance to also sort of leverage that career to go places and see things. You know, it's some people prefer the stability of staying in one place. I like to both go to where the opportunity is. Initially, when I went from Denmark to Germany, it was because the group in Germany had a better opportunity in terms of doing the kind of modeling work that I wanted to do. So I could choose to stay in Denmark and pursue one of the opportunities that were around there, or I could go to where I could learn from those who were good at the things I wanted to become good at.
Yeah.
So I think not being afraid of pursuing the opportunities, even if it requires moving yourself both with and without family, because obviously that becomes more complex as you grow older and you have a family, perhaps that needs to move with you. So, but that's at least gives you the opportunity to go for quality instead of sort of immediate proximity.
Yeah.
And of course, it takes time each time. I mean, you're not just learning a new job; you're also learning a new environment. Maybe having to put your kids in a new school. Yeah, I mean, I did have some degree of advantage from early on that Denmark is sufficiently small that you need to learn other languages anyway, unless you want to stay in Denmark. So we typically learn at least 2 languages in primary school already. You start fairly early with English and then a second language. And I learned German in primary school. I switched to French in high school. So I came out with 4 languages before I left the country already.
That's brilliant.
But to then and to live in the country and live in the language is, of course, a different step again. So yeah,
it's a different level, isn't it?
Yeah.
What was important from a personal perspective to be able to embed yourself, not just within the organisation, but the sort of culture of moving to different countries?
I think again a combination of accepting change and challenge, but also over time learning patience. That things don't come right away, and whether that's professionally or personally, you have to accept some difficulty over a period of time. You have to have some persistence to get through those periods and eventually come out on the other side. And that can be as simple as how long it takes to get comfortable enough in a language to function in it. And in my experience, about 6 months. Once you are not immediately lost in a conversation of 3 people and they just talk to each other instead of including you, that's sort of my personal benchmark. But also meeting new people, establishing a social life takes a while. Just like getting yourself settled in a new role within a company, you might not even be moving company, but you might be taking on a different responsibility or a broader responsibility, and there's learning involved each time. So don't expect to be great at stuff on day one. That's that's my advice.
And that continuous learning element. Do you think the international exposure you've had, do you think it's given you a different perspective to how you apply that to your job, to the science and working with teams?
I mean, there can certainly be very different approaches to how work is done, not necessarily on the technical level, but on the organizational level, on the level of hierarchy, on the interpersonal level. I mean, early on, seeing the difference between a lab in Germany for graduate school and a lab in California for postdoc is already different. I think over time, you kind of tend to pay less attention to it because you're kind of expecting things to be different anyway. It might be more pronounced in the ecosystem around it. It's not necessarily so much on the personal level, but there are different ways of doing things, whether hierarchy is important, whether titles are important, whether the way you approach people is different in different countries, right? But I think fundamentally, in the end, in science, people respect your capabilities, your thinking, your skill set, and as long as you are staying polite, you will learn all the other aspects as you go along. But again, also being flexible enough to adapt to these things is certainly a plus.
Fascinating. Thank you for sharing. I'm always keen to hear, if you were to go back to young Jonas, what piece of advice would you give to him or to people who are early in their scientific careers?
Well, not to be in a rush, and I think I mean early on back in high school, I thought I wanted to be very sort of physics math focused, but then ended up not necessarily enjoying my physics teacher from the first couple of years too much in high school, and actually decided not to pursue the highest level. I don't know if that corresponds to A levels in the US, but you have ABC level in Denmark in high school, or had at the time. It may have changed since then. Decided to stop after the 2 first years and actually switched to French instead, which then became an advantage down the road. But sort of felt that maybe I'd sort of set myself back in terms of what I thought I wanted to do. And then switch more in the biochemistry direction for a period until my first university, and then by coincidence again landed on a project with a supervisor that was a physicist who gave me back the enthusiasm and the taste for the physics, which I then ended up going back to and deciding that chemistry was not so much for me anyway. So sometimes things might seem like a step back, or at least a step away from what you think you want to do, but 10, 15, 20 years down the road, 1 year in a different direction is not a big deal, first of all. Second of all, may not be lost at all, because you might have learned something different. You might have an experience, a skill, something that actually ends up setting you apart or being useful, differentiating you from sort of the standard profile, if you just want to think about it from a career perspective. That you either it becomes useful to you personally, either somebody else sees a use for it. It's not a straight path, and it's not always necessary to be efficiently rushing forward on the short time scale right away. You can learn a lot from many different things.
Yeah, and I hear you talk about coincidences and things that almost happen by luck, but I feel that your mindset, that not being afraid to take risks, wanting to continuously learn and be adaptable, that's almost created those coincidences for you. Would you say that's fair?
Well, I'm not sure. I think I see where you're coming from on that. I wouldn't call it luck. I mean, for me, I'm not I don't really have a lot of belief in destiny or fate or anything like that. When I say coincidence, I see it more in the sort of physics way of randomness. Something happens at a particular time without you having selected it to happen so, right?
Yeah.
But again, like you say, if you're willing to grab the opportunity when it comes along, or if you've put in the effort to be able to grab that opportunity, I think a lot of the time it comes down to that as well. It's between having already done the work to put yourself in that situation, being willing to do more work to take advantage of the situation. Then coincidences become happy coincidences, yes. And then you can call them luck in retrospect. Yeah. But usually it turns into bad luck if you don't do something about it, or if you're not prepared for them. So maybe that's a way to put it.
Yeah. Yeah. Okay. Really interesting. So what's next? What's next for Jonas? What's next for Acousia Therapeutics?
Well, so we're running our phase 2A clinical trial right now, where we're trying to prevent the hearing loss that's typically associated with chemotherapy when it's based on, in particular, cisplatin as a chemotherapy product. It's an old drug that's been around for many years, but it still happens to be one of the most effective chemotherapeutic treatments for many solid tumors. We are specifically testing in testicular cancer and trying to prevent that the testicular cancer patients who are typically up to 99% cured by cisplatin chemotherapy. So it's very effective, but it's also highly ototoxic, so toxic for the ear.
Right. Okay.
It used to be thought that that only in adults, that only about 30% of cancer patients would suffer hearing loss from this kind of chemotherapy, but with the increasingly higher focus on the high frequencies of hearing, and also the ability to clinically reliably measure those high frequencies, it's actually turned out that we are well above 80% incidence of significant hearing loss. And there was just a paper published by the Sanchez Group in Florida this year that showed that even in long-term follow-up, about 70 to 80% of patients still have self-reported hearing problems and clinically measurable relevant hearing problems. So it's also not just a short-term problem,
isn't it? Yeah.
So basically, what we're trying to do is to test our lead drug ACU85 by administering it before each chemotherapy cycle. So this is a local administration. ENT physicians are perfectly capable of making a small injection through the tympanic membrane under local anesthesia into the middle ear space. So not very invasive, especially with local anesthesia. And we've developed a thermal reversible slow release formulation that allows us to do a single injection and get drug exposure for on the order of 2 to 3 weeks after a single injection. And we're using this to then design a very at least in our view, a very elegant study where we're using what's called a split body trial design. So one ear gets the active treatment and the other ear gets the placebo. It's also been used in ophthalmology, so it's not something we invented, but decided that could be useful here because it allows you to do intra-individual control.
It and allows patient recruitment. It must help with that as well.
Yeah, you can essentially go with a smaller amount of patients, a smaller number of patients in your trial, but more importantly, it also helps you control variability because the hearing loss from cisplatin is typically symmetric within patient. So you actually get an intra-individual control. You can both look at how hearing has developed versus the baseline hearing in that ear of the patient, but you can also control versus the opposite ear, which actually allows us to do a fully blinded interim readout. We can see how hearing has evolved in both ears of the patient over time, and we have the, of course, under the assumption that the better ear will be the treated ear, the ability to get an early view on whether things are going in the direction that we anticipate. And we have some very encouraging data from the first patients now that suggests that we can get some quite significant benefits if this holds up over the whole patient population, obviously, and holds up with unblinding. Also, always have to hedge that you have your interpretation until you have it validated. But also really confirms that the ototoxicity, the impact on the hearing is very high in terms of incidence, but also very high in terms of degree of hearing loss on those vulnerable high frequencies. So we're looking to generate an interim blinded readout sometime in Q2 next year, and then we have to, since we're following patients up for 3 months after the last treatment, then so we're doing three cycles of chemotherapy in testicular cancer. So that's 3 rounds of 3 weeks with 1 pre-treatment ahead of each chemotherapy cycle, a follow-up at the end of those 3 cycles, and then a long-term follow-up 3 months after. So we expect to have our top-line data in Q3 next year. And as we discussed in the beginning, we're not really set up as a two-person organization to take things to market. That hasn't been the vision for the company since the inception. So we are in parallel with all of this, working hard to find interested acquirers and educate people on the problem and quite frankly also the business opportunity. It's a big relatively big market compared to what you might think. Already, if you're just looking at cisplatin as a hearing loss problem. So that's the vision for right here and the focus for right here, right now. And obviously, another thing to learn in industry is that you need to communicate. You need to make people aware of what you're doing. It's not about you don't give away all the details, just like you don't give away all the details in basic research before you publish. But you need to especially in a new area like hearing loss, where there's not much that has been done so far. You need to be out and about, communicate about what you're doing, answer questions, explain to people the problem, the approach, the opportunity, and find the right partner or acquirer for taking this all the way forward to patients. And whether that then in an acquisition situation involves me or not, time will tell. Probably depends on the acquirer and the philosophy behind that deal. But otherwise, I'll be looking for the next chance to help take something from the preclinical earlier stage and up to the clinical stage, and keep finding the new challenges and opportunities to impact patients' lives in a positive way.
Well, it's fascinating. Thank you so much for joining us. Wishing you all the best for the data readouts next year. But it sounds that there's some real positive signs that that's going to go well.
Yeah, we are keeping our fingers crossed. It's the strong inflection point in phase 2 when you finally see whether everything you expect based on the strong package you have behind of even getting to there, whether it actually lives up to the expectations. But as I said, early positive signals keep us very confident that it's going to end up in the direction that we want it to, and hopefully it'll make a difference as we go forward.
Well, best of luck. Thank you very much, Jonas.
Thanks a lot, Emma. Thanks for having me.
Thanks for listening. If you enjoyed the episode, please like and subscribe and share it with someone else you think will benefit. And for more podcasts, videos, blogs, and insights, come follow me, Tom Froggatt, on LinkedIn, YouTube, Instagram, Medium, or TikTok. See you next time.